it's my pleasure to introduce the first speaker, Christoph Bock from Vienna. So Christoph's well known for his epigenetics work, and then has been working on single-cell analysis as well, developing some of the interesting methods for analysis of fixed cells and other sort of tough tissues. And so it's my pleasure to introduce him.
Thank you very much for the kind introduction and for inviting me. I will speak about computational work and wet lab work combined, because in our lab this is really integrated and it's very difficult to tease apart what was originally based on a computational method and then developed into an experimental protocol, and where the start was actually from a biological question that then became a computational project. 4 different topics. I will start with how we got into single-cell analysis originally, which was perhaps a bit different than most people. We were concerned with the question of how and why cells react very differently when they are treated with a drug. So essentially preclinical research, all the heterogeneity that you see when even in a dish you are treating your cells chemically, and some of these cells respond very differently than others. And I think this level of heterogeneity that is really fundamental throughout biology and often kind of swept under the carpet in order to get this clean story of gene A regulates gene B, causes disease. This heterogeneity needs to be accounted for to overcome what's sometimes called the reproducibility crisis, in particular in preclinical research, where a lot of these things, when you don't account for heterogeneity, become just very transient and very irreproducible. On the other hand, when you embrace heterogeneity as something that's actually a good thing and not just a nuisance that screws up your experiments, it's a great natural experiment, because all the different cells in your dish or in your tissue, they might get exposure to slightly different drug concentrations. They might be in different stages of cell cycle. They might have different neighboring cells, etc. And if you can measure all of these and integrate this with their transcriptome response, you can do something like a cell state association study where you get a lot of power from this heterogeneity. You can also think about targeting heterogeneity, identifying drugs that might reduce heterogeneity before you go in with your chemotherapy to kill off a cancer, and kind of take away some of the robustness that comes from heterogeneity away from diseased tissue. So one specific example, we were helping, when we first got into single-cell analysis, we were helping the lab of Stefan Kubicek at our institute, trying to find out how you change identity of insulin-producing cell tissue. So there were a few studies that have shown that pancreatic alpha cells can be reprogrammed into pancreatic beta cells that are the insulin-producing cells, and which are very important for diabetes. And these colleagues did a chemical screen, and they found a malaria drug, Artemether, that was thought, or that seemed to somehow change the identity of these cells, not necessarily reprogram them, but change the identity to a level that some of these alpha cells start upregulating insulin. And they wanted to find out how this works, but we couldn't just FACS sort the cells and then treat them, because the tissue is really complex and these cells don't survive as separate human tissues. So what we did is we single-cell dissociated human ex vivo pancreas tissue. So from deceased donors that donate their pancreas tissue, we did single-cell sequencing, and then we get very cleanly separated back then with MARTSIC2, relatively small number of cells, but very clean the different lineages of the human pancreas. And then we can take these pancreatic tissue and treat it in vitro as intact tissue, so keeping as much as possible of the kind of in vivo situation. And then again, single cells dissociate, fix such that nothing further can happen to those cells, and perform single cell analysis, and then assign each of the cells in the treated to those clusters of untreated cells, and then kind of do ex post comparison of the treated versus untreated alpha cells, beta cells, etc. separately. So that is kind of a very, I think, a way that is not really used a lot. So for most people go in and dissociate this tissue, but I think if there's kind of one underlying message of my talk is don't forget about perturbations. And drugs are a very important perturbation, and we can use them in combination with single-cell analysis to see how drugs applied to very complex tissue can give rise to cell type specific responses within that tissue. Then the second direction that brought us to single-cell analysis was obviously epigenetics. My lab has done computational methods development for DNA methylation for many years, and obviously that is also something that profits a lot from single-cell analysis, because if you think about epigenetic landscapes, what's happening in development, these are decisions that are happening on single cells. A single cell decides to either go down a very clean lineage of cellular differentiation as outlined here, or it might get blocked as a leukemia cell, or it kind of transdifferentiate out of this kind of clearly defined lineage. And if we want to understand this at the single cell level, we need to push epigenetic assays to single cell. Nowadays, single cell epigenetics is almost, a lot of people tend to focus on single cell ataxia just because it's easy, whereas single cell DNA methylation is, I think, increasingly falling behind, which is a pity, because that is really what defines kind of the early commitment of cells in very permanent ways of being a blood cell or a skin cell or a heart cell. That's very much encoded in the DNA methylation. So we have focused on it early on and developed protocols that work for single-cell DNA methylation. Disadvantage of these protocols, while they work very nicely and with the right bioinformatics, like region-based bioinformatics to cope with this very sparse data, they don't really scale to thousands and thousands of cells, because you can't really do it on droplets. You have the bisulfite, which is very destructive to cells, and afterwards you need to clean up. So you can't just continue in the same droplet. And that means that still the assays that we have at the moment are on the order of a couple of hundred up to a few thousand of single cell DNA methylation profiles. That is limiting, but it's still you can do useful stuff on it. For example, as part of the International Human Epigenome Consortium and the Blueprint project, we took kind of both low input and single cell sequencing to human hematopoietic cells, FACS sorted out these different cell populations, and then used low input sequencing plus for selected populations also single cell sequencing to try and understand this differentiation hierarchy of the human hematopoietic lineage. And indeed, with kind of machine learning and a network reconstruction approach, we could infer much of the transcriptional regulatory landscape or the epigenetic regulatory landscape underlying normal hematopoietic differentiation. That was mainly a proof of concept. We wanted to establish that both the DNA methylation profiling and the computational methods for reconstructing hematopoietic lineages are powerful and working. And in the hematopoietic system, we knew what to expect. So in a way, it was reassuring to see what we were actually expecting. But this technology, having validated it now, is obviously applicable to tissues where you know much less about what's going on and where you may not even be able to sort out any specific cell types based on FACS markers, but you would have to go in single cell. Again, this is very complementary to single-cell transcriptomics, because transcriptomics really reflects what a cell is doing in that very moment, whereas DNA methylation takes a deep look into a cell's past. What was its lineage history? Where it is coming from? And also to some degree into its future potential. Which genes can this cell upregulate upon challenge, for example? So we've also worked, look deeper in kind of individual tissues. This was a study together with Frederik Geismann, who's now in New York, where we looked at how macrophages kind of populate the mouse body, and we looked not only at gene expression but also at DNA methylation, although unfortunately the methylation data were too late for the paper, so the methylation data are not in the data in the paper, but we've looked at those, and kind of this shows again what I said just now, that while the transcriptome data really reflect this kind of commitment into specific lineages of the different organs, the methylation data keep the identity of being a macrophage at heart, even when you are a microglia cell or a kidney macrophage or a Kupffer cell in the skin. And finally, it provides us a way to look at diseases and see where these differentiation hierarchies go wrong. A very interesting tissue, I don't know if few people have probably heard about it, Langerhans cell histiocytosis is a rare genetic disease. It's essentially perhaps macrophages, perhaps dendritic cells, perhaps some strange progenitor cells forgetting their identity and becoming something very different. Not well understood and very difficult to get samples because it's such a rare disease. So what we took here is essentially the infrastructure that we had validated in the normal hematopoietic analysis to try and see what happens in these lesions. There also you can see a relatively large lesion, 2 large lesions in the brain, so where hematopoietic cells accumulate and form something that is similar to a tumor, but it doesn't have this level of genetic evolution that is happening in a tumor, but rather some kind of differentiation hierarchy that is reflected in gene expression, but also in epigenetic profiles. So essentially, key message here, once you have built the lineages of normal differentiation, you can start looking at what's going wrong in various diseases, and including apparent differentiation hierarchies that appear to be very common, certainly in cancer, but also in some other diseases that have kind of an internal hierarchy in their lesions. Ultimately, kind of from the computational challenge for us is now to put all of these together in quantitative models. So many kind of years ago, when Sui Huang, who's now in Seattle, has started to kind of really take the epigenetic landscape literally and took it as a computational challenge to see how this can be transferred into a mathematical problem. This was very mathematic, very academic exercise. He didn't have the data, no one had the data to really start fitting the models. But now we are in a very different setup, and based on single-cell RNA-seq data, it's already starting to become possible to actually fit these landscapes. But what I'm saying is, what I said earlier and what I repeat again, if we're fitting these purely on RNA-seq data, it's not an epigenetic landscape, it's a transcriptional landscape. And we will probably miss out a lot about what's pre-programmed as kind of potential, which this is supposed to be capturing. So we really need to work hard as a community to make sure that epigenetics is not falling, in particular DNA methylation profiling, is not falling too much behind the progress that we are seeing in single-cell RNA-seq. So I've already kind of brought things a bit over to diseases, and obviously what I consider a major future application of single-cell analysis with major contributions of bioinformatics is kind of disease monitoring and seeing how diseases develop over time and in real patient cohorts. For that disease, we have focused a lot of our research on leukemia, specifically on chronic lymphocytic leukemia, which is a disease of old age, shows typical symptoms of a leukemia, but by far not as aggressive as many of the acute leukemias, to the degree that many patients are diagnosed before, based on a blood count, for example, way before they have any symptoms. It's a very interesting disease to study because it unfolds over 5-10 years, so you can take a lot, really go into in-depth time series. But there's also some patients who progress very rapidly and need almost immediate treatment. On a molecular level, it's a chronoproliferation, accumulation of B cells in the blood, in the bone marrow, other lymphocytic tissues, driven by an overactive, constantly stimulated B cell receptor. So these cells permanently get the signal, kind of divide and do something to prevent kind of an immune attack, kind of a pathogen attack to the body, and that then totally takes over the bone marrow and other lymphocytic organs. There is a drug for this, ibrutinib, a BTK inhibitor that is interfering with a part of the signaling procedure, which works very well. And although it's the kind of starts a very systematic program of how the disease is then being treated, and I will come back to that in a moment. But our first contact or our first application to this disease was that we wanted to understand cell of origin and see how differences in the cell of origin influence, how well we can recover this from epigenetic information. So we took Andre Rendero, who's actually in the room somewhere. Andre? That was his first paper after he joined us as a PhD student. We took a cohort of CLL patients. We did ATAC-seq, back then bulk profiling, and then used machine learning to compare to various clinical features. And what really stood out here was the predictive accuracy of chromatin for the MCLL and UCLL subclasses, which are essentially CLLs derived from an early B-cell progenitor versus a later B-cell progenitor with very different clinical outcomes. Encouraged by kind of the level of information that epigenetic information could take us, we then combined this in a time series, looking what happens when we take this drug, ibrutinib, that I was just mentioning, in order to inhibit this overstimulation of B-cell receptor signaling using flow cytometry, ATAC-seq, and single-cell RNA-seq. Lots of bioinformatics, in this case, Gaussian processes played a major role here, and then also some machine learning later on, and defined essentially the molecular program of steps like downregulation of NF-kappa B binding, lineage transcription factors going down, erosion of CLL cell identity, and then quiescent state. So challenge here, again, Andre's project was to derive what is the computational analysis of a very comprehensive data set to a clear biological model where you can explain to a medical doctor kind of these steps are happening in that particular order. And finally, we collaborated with the neighboring lab, Giulio Saperti-Furga at SEM, who had developed a single-cell chemosensitivity method where they are putting the CLL cells in 384 well plates that are pre-filled with drugs, and then use very high-resolution imaging to see, or automated high-throughput imaging to see how different cell types in this in these plates respond very differently to the drug. And then we collected samples before start of ibrutinib treatment and after the start of ibrutinib treatment, and performed our usual ATAC-seq analysis as well as the single-cell drug sensitivity analysis, and from that prioritized combinations of essentially drugs that modulate pathways relevant to the disease in order to find combination or prioritize combination therapies, how we can help ibrutinib to really get over this kind of constant blocking of the disease, but being very far from an actual cure. So this is a relatively generic method that can be applied to essentially to characterize different types of targeted therapies using combination of single-cell sequencing and bioinformatics on the previous slide, and now also imaging to really see kind of how drugs respond and which drugs might have a stronger or weaker effect, with the ultimate goal of implementing something that's not too dissimilar how drug resistance has been coped with in parts of HIV therapy. So essentially to characterize the patient and in much high resolution and find routes through the kind of epigenetic regulatory landscape that would keep a patient out of the danger zone and kind of moving around in areas where you're not running out of therapeutic options. Finally, in the last few minutes of my talk, I want to briefly mention the CRISPR single-cell sequencing technology, which CropSeq, which was motivated by the fact that from everything I have told you so far, although I kept it for this audience at a very abstract conceptual level, but all of these things produce very interesting targets and individual proteins that we might want to follow up for their potential disease relevance. And obviously, then you can go into small-scale biology, but that gets very limiting in terms of throughput. So what we were looking for is an assay that allowed us to test at least thousands, but ideally tens of thousands of genes or epigenetic variants in a single experiment. CRISPR technology, CRISPR screening is very powerful here in its classical sense of a pool screen, where you take your cells, you infect them with guide RNAs that hit a lot of different genes, and then you apply kind of a selective pressure. Typical, this is cell death or cell proliferation. If you're more advanced, it can be FAC sorting, but then you have to clone your individual marker, GFP reporter, or you have to optimize fixation-based FAC sorting. So that works very nicely if you do cancer studies, and this has been done in 1,000 cell lines for proliferation genes in cancer, but it gets very limiting if you're looking for something that's relatively complex phenotypes. So the alternative is you can keep everything separate, having one guide RNA or one set of guide RNAs targeting all the same gene in per well of a 96 well or 384 well plate, and then you can use any readout that you want, at least if it's sufficiently miniaturized to be feasible from the amount of cells you get in a 96 well plate. So you can do, for example, RNA-seq, you can do TAC-seq, you can do DNA methylation profiling, etc. But throughput is relatively limited. Like, if you'd want to do screens with thousands and thousands of cells, 96 well plates will stack up to pretty big towers doing these type of analysis. So we felt we needed something that combines the throughput of the pooled sequencing with the readout of the array sequencing. And back then, single-cell sequencing had really starting to become, or single-cell RNA sequencing with the introduction of DropSeq was becoming sufficiently high throughput that you could think of using that as your readout for all kinds of screens. Because obviously not everything is transcriptome, but a lot of changes have a transcriptome correlate. So even if you're interested in something that might be happening at the protein level or at the signaling level, many cases it is possible to define a signature that converts this into a transcriptome signature. So if we can apply single-cell sequencing to all the different cells of a pooled screen, we would essentially connect this to the guide RNAs. We would get essentially a CRISPR screen with a single-cell RNA-seq readout. Conceptually, that's easy. You make a CRISPR screen in pooled, and then you do single-cell RNA-seq, and then you can reconstruct all kinds of nice regulatory landscapes and systems. There's one technical problem, though, which is why it took a while until this was actually done, is you need to link the single-cell transcriptomes to the guide RNAs. That's not so easy because single-cell the guide RNAs in CRISPR are under Pol3 promoter, so they never get polyadenylated, and you never see them in a classical single-cell RNA-seq type of experiment. And this is essentially where, for this CropSeq technology that I'm presenting here, the key technological advance was we didn't, just like in PerturbSeq, we didn't use brute force technology to just clone in a separate barcode and then connect everything by, for example, Sanger sequencing, but we exploited a technical detail of how the lentiviruses that we are using are actually integrating into the DNA. So when they come into the DNA, they take a copy of the 3' LTR and move it to the 5' LTR. So after the integration of the standard lentivirus that everyone uses for CRISPR screening, you have 2 copies of this particular region. So we thought, okay, if we place the guide RNA in this region that gets copied, then we will have 1 copy to do the work, to do the CRISPR, the editing, and another copy to play around with to make it visible in CRISPR single-cell sequencing. And that worked quite beautifully and was initially validated. We validated it in T-cell receptor signaling, so we stimulated the cells. We took T cells, Jurkat cells, hit them with a library for a lot of genes involved in T-cell receptor signaling, and then stimulated the cells and left them unstimulated, and by CRISPR single-cell sequencing and some, back then, relatively basic bioinformatics, but a lot more has been done on the bioinformatics of CropSeq. In the meantime, we're able to reconstruct essentially the pathway architecture downstream of the T-cell receptor, which we then validated in classical array screens and found that this correlated very nicely both on the bulk RNA-seq level as with also on the flow cytometry, like protein level, with what we were seeing in separate assays. So that is a technology that is kind of combines the advantages of classical CRISPR sequencing with single-cell readout. Advantage is it moves much of the complexity in the vector. So if you use the CropSeq vector, which is available on Apgene, you don't have to worry about much of the details. You can even use that vector for classical CRISPR screens. So what we often do is we do a classical pooled CRISPR screen, genome-wide or with very large libraries where we couldn't possibly afford always doing the single-cell RNA-seq readout. Do the pooled amplification-based readout, but freeze away some fixed cells based on that, such that we can go back to the very same screen and add single-cell RNA-seq to it if we want at some point, in order to add kind of this additional layer of information. It's also kind of the big advantage over PerturbSeq is that it's compatible with pooled cloning. So it's technically totally no problem to clone genome-wide libraries into CropSeq because you don't have this step of keeping the barcode in sync with the actual guide RNA. It's at the moment cost limiting. Like we would for a genome-wide screen, you would at least need to sequence in the order of 1 million cells, or preferably maybe 5-6 million cells, which at current prices is challenging. The other advantage is because it's kind of one guide RNA that barcodes itself, you don't have this problem of template switching that is kind of haunting PerturbSeq and similar approaches, which again is not too much of a deal if you are working relatively small scale, but the moment you're going for libraries with 50, 100, or even 1,000 target genes, then this becomes actually a major headache and issue. So if you want to try this out, I think this is or kind of talk your biologist colleagues into trying it out. It's actually a relatively straightforward assay. It can be combined with 10x technology, which is, I guess, is the most widely used by now, but also with homebrew approaches. Like initially we did the development of the technology using DropSeq. It's also compatible with SmartSeq 2 if you need you want to start looking at very complex aspects of the transcriptome that you don't get from 10x data. We have made an effort to make it as available as possible, but if you have any questions, just send me an email. We've also sent out a lot of copies of the plasmid, so a lot of labs are using it. So chances are that somewhere near you, someone might already have tried it. And obviously, we are very much committed to developing this technology further. On the one hand, scaling it to genome-wide screens, but on the other hand, taking it to kind of new applications, including we've done very nice screens on primary cells. We're also trying out putting it into in vivo screens, although that's obviously limited by the number or the complexity of the cells that you can put into a mouse. Epigenetic screens could be very interesting, and obviously applications to disease biology. Finally, what is kind of early days yet, but could potentially add a lot of power to crop-seq-style screens is high-throughput single-cell multi-omics technology. So, for example, if we had a high-throughput assay that were able to combine, for example, a metabolomic readout with a single-cell RNA-seq readout, we could do kind of high-throughput CRISPR screens with a metabolomic readout or a proteomic signaling readout. And this is obviously a field that we'll see a lot of development in the next few years. But whenever there's an RNA component in it, it's directly kind of comparable or compatible with crop-seq screens. So that's essentially all, and just kind of one final slide that I like to show is keeping in mind that much of what we are doing is on human primary materials. So behind this human primary material is obviously a human being, oftentimes a patient, sometimes healthy volunteers. And within the Human Cell Atlas and many other contexts, there's a lot of dedication to making data as available as possible, meaning open on the internet. But obviously there are people behind this, and I think we need to talk with these people and see that they will not have harm from making their genetic data available, and that they understand kind of why are we doing this, what this is useful for, etc. And this is, just to give one example, we've done a small pilot project in Vienna where we've reached out to very general population and worked with them and actually put whole genomes online, following the example of the Personal Genome Project of Judge Church in Boston, as a kind of an icebreaker and a start to kind of engage in a public dialogue, what it will mean when everyone is going to sequence almost everything. With that, I would like to conclude and thank all the different people who have contributed, our institution, the funding agencies, and you for your interest and now, obviously, questions.
Thank you, Christoph. Questions?
Thank you, Christoph. Very nice talk and concepts and tools. I was wondering, when you want to reconstruct the epigenetic landscape, right? You mentioned that you have to measure DNA methylation together with transcriptomics. So what else do you have to measure, you think, and what can you leave out, and how do you measure the relative importance of the different aspects, metabolites, histone modifications, DNA methylation, etc.?
So kind of what I typically suggest, like if a biologist comes to me and asks, what shall we do on our system? Oftentimes they come and they have already done RNA-seq to some degree. If not, this is typically the first thing still. Although I firmly believe in epigenetics, it's good to first know which genes are active in your cells. Then if you can do more than one thing, it is typically a good idea to go to the other extreme, which is DNA methylation. So if you have the full spectrum, you think of the genome being super stable and the transcriptome changing very much on the environment changes. If I drink a coffee in the morning, my blood transcriptomes will already be different. DNA methylation is kind of is something that is set very early in development and then for the most part maintained unless the cancer develops. And cancer changes quite dramatically. So if you have RNA-seq as kind of what's happening now and DNA methylation as a look into the past and the future of a cell, that is already covering you quite well for these reconstructions. Obviously, ATAC-seq or ChIP-seq, chipmentation, these type of histone modifications, they provide kind of a very nice intermediate to try and make the connection from very stable commitment to certain lineages over chromatin regulation to transcriptome. So if I were to add more, then I would say after transcriptome and DNA methylation, I would add perhaps ATAC-seq and then one repressive mark, H3K27 trimethylation, and one enhancer-associated mark, for example, K27 acetylation or K4 dimethylation. Obviously, metabolomics is a whole different field, but that's not so important to lineage reconstruction because it's much closer to related to what a cell is doing right now. So it comes even further downstream of the RNA-seq regulation. Although there's important feedback back of the metabolome into the epigenome.
Questions?
Hi, thanks for the great talk. So I was interested in, so what would be, let's say, most promising strategies for designing your guides? Because I mean, that's sort of what you put into your crop-seq. And I mean, of course, it depends on the question, but let's say, I mean, that you can target genes or you can target regulatory elements. And so what's your take on that?
So how we did it originally is like a classical CRISPR knockout screen. And there we use the same guide RNA design principles that you would use in a pooled CRISPR screen. So if you have already an optimized library for your genes of interest for classical pooled CRISPR screens in your freezer, you can just take that, go back to the original stocks and clone this into the crop-seq vector, and you're ready to screen. That would be a CRISPR knockout screen. Obviously, you can also do CRISPR activation screens or CRISPR repression screens or enhancer editing screens or epigenome editing screens, but you would have to obviously put more infrastructure into your cell to allow these screens. So I would think 90% of the people who have yet requested the crop, and that I'm aware of, that have requested the crop-seq plasmid and tried these things out, have worked on CRISPR, like crop-seq knockout screens. Although I think that activation and repression is a great topic for the future, it hasn't really yet become a major push.
Great talk. I like the CLL example. You had your landscape and you said you were actually sort of tracing roots, I think you call it, of patients that are sort of perturbing even, right? I totally agree with you with this perturbation aspect. I don't understand, though, how you first really model the landscape, how you understand then, let's say, average roots of patients, and how you can actually then say how a route along this landscape is changing.
So the good thing we had for the one CLL study, the time series study, is we had 7 different time points of the same patient, or 8 different times. We had 7 patients and up to 8 different time points. So we can see how we have real-time series, not just pseudotemporal ordering, but actual real-time series.
Of distributions, right? Single-cell DNA methylation.
Exactly. Single-cell RNA-seq in this case, plus ATAC-seq. And single-cell DNA methylation, I would love to, but the technology doesn't scale. So for that experiment, it was not possible. And this actual real-time series character, that can get you over this problem of actually finding processes over time. It essentially moves the challenge from because pseudocontemporary ordering is great, but because you need to sample only once and the rest happens in the computer. So you accept certain limitations in order to simplify your experimental setup. Whereas And what we did here is working with very motivated clinicians who were actually able to collect after day 1, after day 2, after day 3, after 1 week, 2 weeks, 4 weeks, etc., take blood from the patient. And this gives us the actual time series that provides then the context where we can start finding these trajectories.
And it would be then a subcell type that would be not doing anything, right? So you would have those trajectories for each subcell type or for a group of cells or for the whole patient on that landscape?
So the advantage here is that all cells, by and large, follow the same program, and they just go through the program at different speeds, and such that we can essentially we are fitting a program while at the same time fitting for each individual cell the time it has progressed along this program, and that we can validate based on the actual time points. So in the simplest version, we could have done a bulk analysis over the time series, from that define the program, and then essentially project single cells onto this bulk trajectory. This is not what we've done, but this would be kind of conceptual way how you can use real time series for calibrating single cell heterogeneous time series.
All right, well, let's thank Christoph once again.